Okay, moving on in our DNA unit lecture, whatever you want to call it. Um, we are getting into the next 2 days physically. How do we take that genetic code that is you and then build you with it? Okay, that's the and I'm using humans as an example, but this is all life as we know it. Maybe someday we'll find life that doesn't have this step, and biology will change forever. But that day is not today. Um, so the next few days, we're gonna be talking about how do you actually, okay, so your DNA is your code, that is you, those are your genetics. How do you physically create a person with that or a being? All right. And the answer here is going to be we had to start off with proteins. Okay, so proteins are made by the cell, and in particular, they're made by DNA. So, what a protein is, now this is a lot. I would not write all this down. All right, this is kind of more for me here. But your body is made up of trillions of cells. We know this. Those cells are all different kinds of cells. So we have muscle cells, brain cells, blood cells, and more. All right. Inside all of those cells and around all of those cells are proteins. So proteins are allowing your body to do what it does. It breaks down your food. It helps power your muscles, sends signals to your brain, controls the body, transports nutrients to the blood. So essentially, proteins are the building blocks of life. All right. Um, yeah, I'll leave it at that. All right. Proteins are the building blocks that make up what you are. And I got a pile of Legos there. So just like when you're building with Legos, you have the instruction manual, that's your DNA. And then the proteins are actually what it's building with. All right. Now some of those things are obvious, like skin color, eye color, what kind of hair you have, your height, those kind of things. But a lot of those proteins are acting on the cellular level. Okay, they're breaking down the food that you eat. They're fighting disease. They're going through and catalyzing cell processes. All right, so a lot of them are just the vast majority of your DNA is just basic human function. All right, there's that last like 1 to 2% of your DNA that's like, oh, what makes you unique? But the rest is just like, no, this is the DNA you need to be a human being kind of thing. Okay. So goal for the day is we're going to turn, I'm going to break it into 2 days. All right. As we're going to turn DNA into proteins because DNA is the instructions. Proteins are the Legos and the building blocks. How do we get from one to the other? Transcription, I believe that link is broken, so we won't go there. So transcription is the first step. All right. One of the strands of DNA is going to be used as a template to create a strand of RNA. And that RNA is more specifically mRNA or messenger RNA. Okay, so wherever that gene is located on the DNA, and there's a lot of, once again, we're getting the sophomore high school version of this, but there's a lot of markers along the way that say, hey, this is the start of this gene, etc. But we're going to use one side as our template to create the complementary strand of RNA. And it requires this big triangular looking enzyme called RNA polymerase. So RNA polymerase, when we replicated DNA, we had a bunch of different enzymes, right? With transcription, really it's kind of a one-man show. There's some other helpers, but really it's RNA polymerase that is going to unzip that gene, build the strand of mRNA, trailing it, and then zip the gene back together there. Okay. It is going to always start. So how does it know, your DNA is 3 billion letters long. How does it know where to start? It always starts at a particular region. It's a series of A's and T's that lets it know, oh, here's the start of a gene. And then we call that region the promoter. So it chemically is attracted to the promoter, and it will wind through all that DNA till it hits the end, and then it'll kick out. All right. Here are the meat and potatoes of the transcription process here. Okay, so those chunks of DNA that we're going to be getting genes from are called exons. All right, exons as in they are expressed. All right, so not all your DNA has genes. The sketch down below here. All right, there's a lot of stuff in your DNA. All right, there's a lot of things in there. but there's only some chunks of it that actually code for genes. The part that we need to be able to understand is number 5 here. Okay, every 3 bases on mRNA is a codon, and it is going to respond to a particular amino acid. Okay, so here is what's called a codon chart. And this is real. Okay, that is the real, that's not a watered down version. If you have a sequence that is CCU, we see that here, it will code for the amino acid proline. That is the building block of the protein. And so every 3 letters is a new piece that's going to be added. And based on what pieces are added and in what order they're added, it's going to shape and build a particular protein, just like we would with Legos. Okay, so what we need to know is that it starts the promoter. It uses RNA polymerase. C bonds with G, A bonds with T, unless it's on the RNA, then it's a U. Tricky that way, but that's life. And then every 3 letters are going to code for one particular amino acid. All right, that's going to be huge when we take our test. So we'll get a little bit of practice with that today, but just getting used to 3 letters is one piece of the protein as we're going. I'm actually going to stop right there. We're just doing transcription today. We're going to keep it nice and simple, nice and concise before we add translation on another time. So there you go. A little DNA transcription today.